Okay, so good evening. I'll start today's session. I'll share my screen and we can see if I'm visible and if I'm audible. Give me a minute. Yeah, so I've shared my screen. I hope I'm audible and my screen is visible. Okay, so I'll start today's session. So today is the sixth live session of this course, NPTEL Class RNA Biology. So as you know that in this course we basically discuss some assignment questions of last year, and we basically try to discuss the concept behind these questions so that it's easier for us to not only solve this question but similar type of other questions as well. Okay, so that is the basic concept of these live sessions, and we try to keep the sessions as interactive as possible. So if you want to say something, you can just write it in the chat box or raise a hand or unmute and just let it out. So we'll start with the first question for today. Let me put this on slideshow. So the first question for today is, exporting T function depends on, these are the four options: RNA quantity inside nucleus, RAN GTP, RAN GAP, or exportin 5. So before we move on to discussing the question and basically the answer, the concept behind the question, I'll give you 2-3 minutes to ponder upon it, and if you can answer, that's great. Then we'll move on to the question. So the right answer for this will be RNA-GTP. So what happens during nuclear cytoplasmic exchange is that the transportation between cytoplasm and the nucleoplasm, which is critical for survival of all cells. So it is a very tightly regulated process. Okay, and several million macromolecules selectively pass through nucleus and cytoplasm every minute. Right. So nuclear envelope is a physical barrier which regulates the traffic between nucleoplasm and cytoplasm. So it is a phospholipid bilayer membrane which consists of two layers, inner and outer membrane. So the inner and outer membranes are separated by the perinuclear space, and the cytoplasm is connected to the nucleoplasm via nuclear pores. So although small size of molecules, they freely move through the nuclear pore, bigger molecules need the help of special carrier proteins. Right? So what happens is that this X protein T is a protein that helps transport mature tRNA molecules out of the nucleus. It's also known as export. So X protein T binds to tRNA with high affinity in the presence of RNA-GTP. It shuttles rapidly between the nucleus and cytoplasm. It interacts with nuclear pore complexes. It binds tRNA directly and with high affinity, and it's found predominantly in the nuclear region. It's a carrier protein that belongs to the karyofurin family. It mediates nuclear export of RNA, tRNA. It's specific mediator of tRNA export. It binds and exports mature 5' and 3' end processed tRNA molecules out of the nucleus. So it acts pleiotrophically in plant development. In particular, pause 5 and pause 6 result in delayed leaf formation during vegetative development, and it's a potential therapeutic and prognostic marker. Right? This is all about the background of exporting. Okay, so it is a nuclear export receptor that binds to RNA-GTP and tRNAs to export them out of the nucleus, right? So this process is important for the maturation of tRNAs in the eukaryotes. So how does XPOT and RNA-GTP work? So XPOT binds to RNA-GTP and mature tRNAs to form a nuclear export complex. Now this complex shuttles to nuclear pore complexes, NPCs. Now the interaction between XPOT and tRNA is enhanced by the RNA-GTP binding. Now RNA-GAP1 and RNA-BP1 disassemble the complex, releasing tRNA into cytoplasm. XPOT is a carrier protein that belongs to the karyofurin family. It shares similar architecture with other important-like proteins. So XPOT is mainly nuclear at steady state but shuttles bidirectionally through NPCs. So what does this process mean? XPOT is responsible for the majority of tRNA export in xenopus oocytes. It is a specific mediator of tRNA export and shuttles rapidly between nucleus and cytoplasm. Right? So transportation of macromolecules including protein or RNAs between nucleoplasm and cytoplasm occurs through nuclear pore complex in the nuclear envelope. Now NPC is highly selective and bidirectional transporter for various cargo molecules. There are 4 important factors for the nuclear transport: NPCs and nucleoporins, nuclear localization signals and nuclear export signals in cargo molecules, RAN proteins that allows for active transport and bidirectionality, and karyofurin like importing, exporting, transporting that can recognize cargo molecules. Let's talk about the nuclear pore complex and nucleopore. So NPC is a complex basket-like structure with huge molecular mass of 120 megadalton in humans. It is made up of NUPs with 100 to 150 nanometer in diameter and 50 to 70 nanometer in thickness according to the species. So each NPC has a central role, 30 nanometer in diameter and around 50 nanometer in length, that connects between the nucleoplasm and cytoplasm. So the main structures of NPC include the inner pore ring, the nuclear and cytoplasmic rings, the nuclear basket and cytoplasmic basket. Now, NPC is composed of multiple copies of 30 to 50 different NUPs, and 500 to 1000 NUPs are integrated into the NPC structure. So NUPs serve as an additional architectural scaffold and a permeability regulator. Some NUPs have intrinsically disordered domains rich in repeating amino acid sequences such as FXFG, phenyl X-phenyl glycine, phenyl glycine, or glycine, leucine, phenyl alanine glycine, which act as docking sites for carrier-ferrets, like important or exported. So these repeating sequences line the central hole and regulate the passage of cargo molecules, such as NUP62 complex, which consists of NUP62, NUP58 and NUP54. They have been shown to include FG repeats. Now, what is the nuclear localization signal? Nuclear localization signals, NLS, or nuclear export signals, NES, the proteins required for nuclear functions such as DNA polymerase, RNA polymerase, transcription factors, and many others are targeted to the nucleus and specific amino acid sequences called NLS. The NLS is recognized by nuclear transport receptors that direct the transport of the proteins to the NPCs. So the first NLS was characterized by Alan Smith and colleagues in 1984 from simian virus SV40 T antigen as a classical monopartite sequence. So classical NLSs contain monopartite or bipartite signals. Now bipartite NLSs have two clusters of basic amino acid sequences. The prototypical bipartite NLS is nucleoplasmin found in Xenopus laevis. Now, if you talk about nuclear export signals, NES, some proteins remain within the nucleus following their import from the cytoplasm, but many others shuttle back and forth between the nucleus and the cytoplasm. So the proteins are targeted for export from the nucleus by specific amino acid sequences called nuclear export signals, and the NES contains hydrophilic amino acids. The consensus sequence for NES was this, and so basically this particular symbol represents hydrophobic residues. Now NES is recognized by receptors within the nucleus called exportin, which directs protein transport through the NPC to the cytoplasm. And different exportins have their specific cargo molecules. For example, CAS or exportin to transports in protein A. CRM1 exportin 1 is a ubiquitous nucleic export transcriptor containing hydrophobic residues. So this is an example of nuclear localization signal. This is how it works. So carrier-ferrets, also known as importins or exportins, are a superfamily of nuclear transport receptors that facilitate the translocation of proteins, RNAs, and ribonuclear particles across the NPC in a RAN-GTP hydrolase dependent process. So more than 20 carrier-ferrets in human have been reported. Among them, 11 carrier-ferrets, importin beta, importin beta 2, importin 4, importin 5, and importin 7, importin 8, importin 9, importin 11, transportin SR, importin 13, exportin 4 are involved in import or bidirectional transport of cargo molecules. 8 exportins like exportin 1, exportin 7, exportin T are involved in the export of cargo molecules. Now, each carioferin has its own specific cargo molecules. For example, exportin 2 contributes to the export of importin alpha and exportin K to the export of tRNA. And carioferins can also export nucleotide motif including tRNA, miRNA, rRNA, viral RNA and uridine-rich small nuclear RNAs. However, structure of mRNAs is highly diverse unlike other RNAs, so they can be transported by processing and assembly into ribonucleoproteins. RAN proteins. The activity of carioferins are regulated by interaction of a protein called RAN. RAN gives the nuclear transport bidirectionality through the nuclear pore. So RAN is a GTP binding protein whose conformation and activity is regulated by GTP binding and hydrolysis. RAN GTP is about 100-fold more abundant in the nucleoplasm than in the cytoplasm, which is possible due to the fact that RAN's guanine exchange factor RAN-GEF is located in the nucleoplasm. Similarly, the RAN-GTPase activating protein RAN-GAP is located in cytoplasm. So the activity of RAN-GEF and RAN-GAP ensures the unequal distribution of RAN-GTP across the nuclear envelope with high concentration of RAN-GTP in the nucleus, which provides directionality of transport across nuclear envelope. So if we see, this is the distribution of RAN-GTP across the nuclear envelope. Now, what is the import cycle? The first step of the nuclear transport cycle is the formation of important cargo complex. So, importants can bind cargo molecules after recognition of their NLS. Depending on types of NLS, different importants are involved. After the formation of importance cargo complex, important is specifically recruited to NPC in the nuclear pore, and then the complex can pass through the nuclear pore. In the nucleoplasm, binding of RAN-GTP to important cargo complex facilitates distribution of cargo, and then RAN-GTP bound importance exported to cytoplasm. Recycling of important RAN-GTP complex is mediated by nuclear export receptors. Cytoplasm GTP is hydrolyzed to GTP. This releases the importance so that it can bind to a new cargo protein in the complex and participate in the another round of transport. The protein import through nuclear pore complex. Now the export cycle. Cargoes containing NES are recognized by exportins. The NES bound exportin forms a complex with RAN-GTP. RAN-GTP is 100-fold more abundant in the nucleoplasm than in cytoplasm. Binding of RAN-GTP to exportins induces high affinity of exportin for its cargo molecule. The exportin cargo RAN-GTP complex basically binds the docking site of NPC and passes through nuclear pore. So RAN gap in the cytoplasm hydrolyzes RAN-GTP in the complex, which leads to the association of the complex. Now, regulation of nuclear protein import. The transport of proteins to the nucleus is highly regulated event. Such as transcription factors, import and export into the nucleus is a well-established mechanism to control these process protein activities. Since one such mechanism, TFs associate with cytoplasmic protein that mask their NLS and therefore they remain in cytoplasm. For example, under unstimulated condition, NF-kappa B binds with I-kappa B inhibitory protein in cytoplasm. I-kappa B masks NF-kappa B NLS and it remains in cytoplasm. In stimulated condition, I-kappa B is phosphorylated and degraded, allowing NF-kappa B to enter nucleus and activate transcription of target genes. Another example is of transcription factor PHO4, This analysis is masked by phosphorylation. When this transcription factor is required inside the nucleus, this phosphorylation happens and it's imported into the nucleus. Now, all RNAs are transported across the nuclear envelope as ribonuclear protein complexes. The importins and exportins transport tRNAs, RNAs, miRNAs, and snRNAs in an GTP dependent manner. So, RNAs associate with ribosomal proteins and other factors in nucleolus and exported to the cytosol by CRM1. Now, tRNA and miRNA precursors are exported T and exportin 5 respectively. So, mRNA are exported by a distinct mechanism that does not involve carriopharynx and is independent of RAN. Pre-mRNAs are associated exporter complex in the muscle in concert with the completion of splicing and polynylation. Exporter complex directs mRNAs through the NPCs. Reactionality of the process is established where RNA helicase localized at the cytoplasm phase of the NPC. The mRNA exporter complex is remodeled by helicase as it reaches the cytoplasmic side of the pore. This releases the mRNA into the cytoplasm and prevents its transport back into the nucleus. Now, many small such as RNAs such as snRNAs and slow RNAs function within the nucleus as a component of RNA processing machinery. snRNAs are tissue transported nucleus cytoplasm where they sit with proteins to form snRNPs and then returns to the nucleus. So CRM1 and other transport receptor protein that binds 5,7-methyl guanosine caps of snRNA is involved in the export of snRNA to cytoplasm. So in contrast, the sequences present in the snRNP proteins are responsible for the transport of snRNPs from cytoplasm to nucleus. Now the nucleus contains most of the cell's fate, allowing for sophisticated regulation of green gene expression. Similarly, an envelope is a double membrane that surrounds the vapor. The nucleus contains compartments that do not make any sense at all. That are not membrane bound. The nucleus contains compartments that are not membrane bound. The nuclear envelope contains pores used for importing proteins to the nucleus and exporting RNAs and proteins from the nucleus. So, nuclei vary in shape, in size from about 1 micron to more than 10 micron in diameter. Most cells have a single nucleus, but some cells contain multiple nuclei and a few cell types lack nuclei. The percentage of the genome that is heterochromatin varies among cells and increases as cells become more differentiated. Now, chromosomes occupy distinct territories. Although the nucleus lacks internal membranes, nuclei are highly organized and contain many subcompartments. Each chromosome occupies a distinct region of territory. This prevents chromosomes from becoming entangled with one another. So, the nucleus contains both chromosome domains and interchromosomal regions. Now, the nucleus contains subcompartments that are not membrane bounded. So, nuclear compartments are not membrane bounded. RNA synthesized and cytoplasm RNA synthesized and ribosomal subunits are assembled in the nucleolus. The nucleolus contains DNA that encodes RNAs and is present on the multiple chromosomes. mRNA splicing factors are stored in nuclear envelopes. They move to steps of slides of transcription where they function. So other nuclei bodies can be identified with antibodies, but the functions of most of them are unknown. So nucleus is totally bounded by the nuclear envelope. It's surrounded by a nuclear envelope consisting of two complete membranes. The outer membrane is continuous with the membranes that of the endoplasmic reticulum. The lumen of the inner membrane is continuous with the lumen of the ER. The nuclear envelope contains numerous NPCs. They are only the channels in the transport and molecules and fine micromolecules between them, nucleus and cytoplasm. Now, the nuclear lamina underlies the nuclear envelope. So, nuclear lamina is constructed of intermediate filament proteins called lamins. The nuclear lamina is located beneath the inner leaf nuclear membrane, and they are physically connected by stamina-associated integral membrane properties. So, nuclear envelope plays a critical role in the nuclear envelope assembly, and they may provide physical support for the nuclear envelope. The proteins that connect the nuclear lamina to cartoon. This may allow the nuclear lamina to organize. This may allow the nuclear lamina to organize DNA replication and transcription. Uncharged molecules smaller than 100 daltons can pass through the membranes of the nuclear envelope. Molecules and macromolecules larger than 100 daltons across the nuclear envelope may bonding through NPCs. Now, particles up to 9 nanometer in diameter and can pass through NPCs by passive diffusion. Larger macromolecules are actively transported through NPCs and must contain specific information in order to be transported. Now, nuclear pore envelope are symmetrical structures that are bound at side where the inner and the outer shell membranes are fused. Each NPC is given this as a mass of 120 daltons, 4 times that of a ribosome. So, it is constructed from minute copies of DNA. It is introduction. NPCs contain fibrils that extend the cytoplasm and vascular structure that extends the nucleus. Protein organization in a nuclear envelope. So the protein of NPCs are all called nuclear proteins. Many nuclear proteins contain repeats of short sequences which are thought to interact with transport factors during transport. Now, some nuclear proteins are transmembrane proteins that are thought to anchor NPCs in the nuclear envelope. All of the nuclear proteins of each NPCs have been identified. NPCs are disassembled and reassembled during mitosis. Some nuclear proteins are dynamic. They rapidly sit with and dissociate from NPCs. Now, mature nuclear proteins contain sequence information required for the nuclear localization. Proteins selectively for enter the cell and exit the nucleus through the nuclear ports. Transmit for a nuclear import lies in a small portion of the transported protein. A nuclear localization sequence is often a stretch of basic amino acids. They are defined and are both unnecessary and showing unnecessary in showing for nuclear import. The receptors for nuclear import are cytoplasmic proteins that bind to the NLS or cargo proteins, and nuclear promoter receptors are part of the large family of proteins often called carrier-ferrins. The most essential amino acids which in leucine act as the most commonly nuclear envelope exposed sequences. So, what are the things that nuclear envelope wants? Bites that proteins that can contain nuclear exposed sequences. And in the nucleus, transportation is cytoplasm. Now, the RAN is a small GTPase that is commonly in all eukaryotes and is found in both the male and the cytoplasm and the nucleus. The RAN GTP promotes hydrolysis of GTP by R. The RAN GA promotes exchange of GDP from GDP for GDP on RAN. RAN gradient through between the cytoplasm and ectoplasm. In the cytoplasm, the energy needed to create this gradient is supplied by 120 ATP, transforming RAN GDP to RAN GDP. Thus, the nucleoplasm is sink of RAN GDP and the source of RAN GDP. In the cytoplasm, RAN GDP is converted into RAN GDP. Thus, a sink of RNA is in this way two gradients are created, RAN GDP and RAN GDP. The icons the figure depicts levels of concentration. So molecular trafficking across the nuclear pore are complex mediated by karyotype variants and importance and exportance. So the RAN gap is cytoplasmic. There is the RAN GEF is located in the nucleus. So RAN controls nuclear transport by binding carrier and affecting the ability to bind the carbons. So DNA is packed with proteins to not condense. That we all know. Now, chromosomes are different regions with different functions. Chromatin densely packed centromere cell regions, chromatin less densely packed regions lacking transfer genes. So, if this is a variant of different sources and like SH3.3, it improves in the size outside of the S active transcribed regions, all of this. So coming back to the question that we started with, exporting T functions depends on the RAN GTP, right? So if anybody has any question, they are free to raise their hands and unmute and let me know, or move on to the next question after waiting for 2-3 minutes. No, there's no doubt in this question. We move on to the next question for today. Transposons play a role in these were the four options: exon deletion, intron deletion, exon gain, and intron gain. I'll wait for a few minutes. If you can answer this question, then I'll move on to the discussion. So the answer for this will be intron gain. So first we'll discuss what is transposon basically, right? So transposon is a DNA segment that can move to different locations in a cell's genome. So they are also known as jumping genes, and they move by a cut and paste mechanism. So the transposase enzyme recognizes specific sequences on the transposon and cuts it out, and the transposon is then reintegrated into a new location in the genome, and the target site DNA is duplicated, creating a target site duplication. So they can cause mutations, they can increase or decrease the amount of DNA in the genome, they can transmit antibiotic resistance among bacteria, they can be used as non-viral gene editing methods, etc. So American scientist Barbara McClintock discovered transposons in 1940s and 50s. Right. So if you talk about DNA recombination, it is homologous recombination. It's precision. Like HR mediates exchange between DNA segments that share extensive sequence homology. An exchange may can occur at any point between the homologous region, although particular DNA sequences may influence the frequency of exchange. Now, efficiency. Whenever sufficiently long homologous sequences are brought together in a single cell under appropriate conditions, the production of recombinant sequence is more of a rule than exception, right? So, several proteins are required in base pairing and strand exchange. It's a conserved cellular process. Extensive homology is not required, limited to specific sites, requires sister-specific proteins, and is independent of homologous recombination. Now, if you talk about site-specific recombination, it's a conservative SSR. Recombination occurs at specific sites within short sequences identity. Strand exchange occurs by precise breakage and joining events. No synthesis or loss of DNA sequences occur. Recombination is reciprocal, so non-conservative or transpositional. Like no homology between the recombination sites. DNA synthesis accompanies breakage and joining events. Recombination is non-reciprocal. It's non-reciprocal. Now, what do you mean by non-conservative or transpositional? Okay, so no homology between the recognition and recombination sites. DNA synthesis occurs. DNA synthesis occurs accompanies breakage and joining events, and recombination is non-reciprocal. Right. So this is, you can see the biological consequences. Sorry, of yeah. So what are the biological consequences of recombination? Stable plasmid maintenance requires that plasmid copies efficiently distribute into daughter cells at cell division. So high copy plasmids are randomly distributed. Low copy plasmids have to use specialized partitioning system. Now both mechanisms require that the presence of multimeric plasmids, which can be generated by homologous recombination, and these multimers must be minimized to monomers prior to partitioning. So P1 fudge uses site-specific recombination system pre-locks to resolve multimers into monomers. Single protein bearing recombinase activity Cre is required for the reversible recombination reaction. Okay. So we'll not go into the too much in intricate details. So what are the features of transposons? Autonomous may have a long non-autonomous form, excised non-replicatively and reinsert elsewhere. Okay. So means activator element. DS elements, simple DS elements are deletion derivatives of AC which have lost internal sequences for transacting factors. So I'll not go into much detail of all of these. So this is how you can explain transposon. Components not drawn proportionally. Terminal inverted repeats 31 base pairs in length. Encoding transposase protein. This is a transposon. So there were class 1 and class 2 transposons. Transcriptional RNA reverse transcript is using reverse transcript is encoded by transposon and cut and paste mechanism for class 2. For class 1 is original transposon never leaves DNA and class 2 transposon leaves original location to reinsert. So the first step will be accession, transposes gene transcribed and translated, drift transposes enzymes and integration. So what is the transposon activity? Steps of transposition, accession. Number 1 will be accession, right? So accession is transposase binds to terminal inverted repeats, dimerizes, cleaves transposon out of donor site. Drift transposase carries transposon to target site. And number 3 is integration. Transposase inserts transposon into target recipient site. So what is disrupting transposition? Changes to transposase gene after inverted terminal repeat sequence can interfere with transposition. So disrupting transposition is changes to transposase genes, as I said. So inverted terminal repeat gene transposase cannot recognize where to bind to excise transposon. Transposase gene mutation. Okay. The transposon is stuck in DNA. Inverted terminal repeat change. Transposase cannot recognize where to bind to excise transposon. So transposase gene mutation, non-functional transposase cannot excise transposon. Now if you talk about autonomous transposon with intact inverted terminal repeats and transposase gene, transposon can be moved without outside help. A non-autonomous is transposon with intact inverted terminal repeats but non-functional transposase gene. Transposase enzyme must be added from somewhere else. So where are transposons found? In almost all organisms. 50% human genome sequence is transposon based. Transposition causes mutations in genome which can be harmful to the organism but also leads to genetic variation which can lead to evolution. Now where are transposons found? In almost all organisms. Transposition causes mutations in genome which can be harmful to the organism but also leads to genetic variation which can lead to evolution. Now what are transposable elements? The general features of transposable elements, prokaryotic transposable element. Transposable element is mobile genetic element of a chromosome that have the capacity to move from one location to another in the genome. Normal and universal components of prokaryote and eukaryote genomes. Prokaryotes transpose to or from cells chromosome, plasmid or a Faj chromosome. Eukaryotes transpose to and from same or different chromosome. What are the non-homologous combination? Transposable elements insert into DNA that has no sequence homology with the transposon. And transposable elements cause genetic changes and make important contributions to the evolution of genomes. Like insert into genes, insert into regulatory sequences, modify gene expression, and produce chromosomal mutations. If we talk about transposable elements, two classes of transposable elements or mechanisms of movement encode proteins that one move DNA directly to a new position or to replicate DNA and integrate replicated DNA elsewhere in the genome, like prokaryotes and eukaryotes. Electrotransposons encode reverse transcriptase and make DNA copies of RNA transcripts. New DNA copies integrate at different sites, eukaryotes only. Insertion sequences, elements, transposons. Now, what are insertion sequences or elements? Simplest type of transposable element found in bacterial chromosomes and plasmids. Encode genes or transposase for mobilization and reinsertion. Range in size from 768 BP to 5 kb. IS1 first identified in E. coli's galactosoperon is 768 base pairs long and is present with 4 to 19 copies in the E. coli chromosome. Ends of all non-IS elements show inverted terminal repeats. The integration of an IS element may disrupt coding sequences or regulatory regions, alter expression of nearby genes, cause deletions and inversions in adjacent DNA, resulting crossing over. The original copy remains in place, new copy inserts randomly. So transposition requires transposase coded by the IS element. IS element otherwise uses host enzymes for replication. Transposition initiates when transposase recognizes ITRs and site of integration will be target site. Now, staggered cuts are made in DNA at target site by transposase. IS element inserts, DNA polymerase and ligase fill the gaps. Transposase behaves like a restriction enzyme. So, small direct repeats flanking the target site are created. The integration of IS element in chromosomal DNA. Similar to IS elements, but are more complex structurally and carry additional genes. Two types of transposons: composite transposons, non-composite transposons. So, composite transposons carry genes. Example might be a gene for antibiotic resistance, flanked on both sides by IS elements. Non-composite transposons, they carry genes. Examples like might be for antibiotic resistance but do not terminate with IS elements. Okay. Barbara McClintock, Cold Spring Harbor, New York, Nobel Prize in Physiology and Medicine for her discovery of mobile genetic elements. He studied transposable elements in corn, formally identified as mutated genes by Marcus Rhodes, also known for work demonstrating crossing over as part of the chromosomal basis of inheritance. So, what are the general properties of plant transposons? Possess ITR sequences and generate short repeats at target sites, may activate or repress target genes, cause chromosome mutations, and disrupt genes. These two types, autonomous elements transpose themselves, possess transposition gene. Non-autonomous elements do not transpose themselves, lack transposition gene, and rely on presence of another. McClintock demonstrated purple spots in otherwise white corn. kernels are results of transposable elements. Clintock's discovery of transposons in corn. So, transposition effects on corn kernel color. Will not go into too much detail. What are human transposons? They are flanked by 7 to 20 break repeats. Some are transcribed, thought to be moved by RNA intermediate and all of that. This is the composition of the human genome, exons, DNA transposon, retrotransposons, etc. So this is, if we talk about transposable elements, there can be retrotransposons or DNA transposons. Copy and paste mechanism, replicate mechanism, cut and paste mechanism. So what are the applications? Plant molecular biology, identifying cancerous genes. Transposons play a role in intron gain, right? So the answer, coming back to the question that we started with, that transposons play a role in intron gain. Like how? As research suggests that many new introns in eukaryotic genomes originate from the insertion of transposable elements, which, if positioned appropriately, can be interpreted as introns by the splicing machinery, leading to the creation of new introns within a gene. Okay. So, what is the mechanism? When a transposon inserts itself into a coding region of a gene, if the insertion happens to contain splice site sequences, it can be recognized by the splicing machinery and removed during RNA splicing, effectively acting as a new intron. Some studies have identified specific types of transposons called introners, which appear to be particularly efficient at generating new introns due to their structural insertion patterns. This mechanism is thought to be a major contributor to the diversity of intron structures across different species, allowing for rapid intron gain in certain lineages. So, according to transposon hypothesis, introns originate as transposon insertions, as in the case of new rise intron. New insertion either serendipitously possesses or quickly acquires signals, allowing it to be efficiently spliced out of transcripts. So the correct answer will be intron gain. So I'll wait here for a few minutes. If you have any doubts, then I'll move on to the next question. So we'll move to the next question for today. Histone unique to bar body, and these are the four options. Sorry. So I'll give you 2 minutes to tell me which one might be the right answer, then we'll discuss. Okay, so the answer will be H2A 1.2. So what is a bar body? A bar body is the inactive X chromosome in a female somatic cell. Okay, so this is known as X chromatin or bar body after the name of its discoverer Murray Bar in 1940. It's inactivated condensed X chromosome found in female cells. X chromosome inactivation is random and occurs at an early point of development. Now, what is the need for a bar body? So, to regulate the amount of X gene product, that is dosage compensation, and in sex determination mechanism, the female have XX chromosomes while the male just have one. To compensate this, one of the X chromosomes is inactive in females. So, X chromosome inactivation is random and occurs early in embryonic life and remain fixed. Now, where is this bar body found? It can be seen clearly during interphase. It's found in the nucleus of somatic cells in female, and it's commonly situated at the periphery of the nucleus. So in nerve cells, it may be near the nucleus or near the nuclear envelope. So in buccal epithelial cells, it's generally attached to the nuclear envelope. In neutrophils, it may appear as a small rod called drumstick. So this is the bar body in the nucleus. The positive bar body indicates female sex. Okay. So what is the frequency of occurrence in nervous tissue? The frequency may be 58.5%. For chorionic epithelium, it may be as high as 96%, and in oral space, it may vary between 20 to 50%. Now, what are the reasons for bar body test? To identify, process gender, and to establish sexual identity in newborns and adults. As a screening test for ambiguous genitalia. Delayed onset of puberty, times in infection. What is the aim? To identify the presence of bar body in the female buccal cavity. Buccal epithelial cells especially have bar body structure, which are considered to play a major role for sex determination. So this small round bar body is located either in the border of nuclear membrane or sometimes inside of the nucleus. So this bar body may be single or more in number in some cases, and these structures are present only in the female sex. We'll not go into this part. So if you can see, this is a human karyotype. This X and Y chromosome, basically that determines if it's a male or a female, right? So if we see, this inactivation takes place in the early embryo. Random selection of the woman is mosaic of clones with the maternal or the paternal X active. Random selection in each cell of the X that remains active. Each cell generates a clone with the same X active. X inactivation is an epigenetic process. Because of X inactivation, every female is a mosaic of cell lines with different active X chromosomes. So X inactivation, dosage compensation, and the expression of X-linked genes. Same amount of X-linked gene products between males and females. Males with one X chromosome, females one two X chromosomes, and yet the mean amounts of gene products of X-linked genes are the same in male as in females through X chromosome inactivation. So what is the X chromosome inactivation mechanism of X chromosome inactivation? XIC. XIC controls expression of the XIST gene. XIST X inactivates specific transcript. XIST produces a non-coding 17 kb RNA molecule. It codes the entire local X chromosome cis-active. Okay. So it's located in XIC transcript only from the inactive X. So how XIST silences the future inactive X expressed from the future XI? Both the chromosome establishes the inactive state. Only X, non-X is active. The XIC region of the human X chromosome is this. So what is the molecular mechanism behind X inactivation? The key player is the X-linked gene XIST. X inactivates specific transcript chromosome XQ13.2. So XIST starts trying to produce a non-coding RNA that codes the X chromosome and inactivates it. XIST is uniquely expressed from the inactive X. XIST RNA does not travel over to any other X chromosome in the nucleus. That is 6-axis action. Bar bodies are inactive X chromosomes painted with XIST RNA. And XIST is the master regulator for X inactivation. It is expressed only from the allele on the inactive X. It is transcriptionally silent on the active X in both male and female cells have a function in the initiation phase of X inactivation. A transcription of XIST ceases on the other X chromosome, allowing all of its hundreds of genes to be expressed. Shutdown of the XIST locus on the active X chromosome is done by methylating XIST regulatory sequences. So methylation permanently blocks XIST expression and permits the continued expression of all the other X-linked genes. So the process is XIST pronounced exist, includes a large RNA molecule, codes XI from the XIC near the centromere outward along the X chromosome. So what are the mechanisms for combating XI? Bar body enzymes cause the following to occur: high levels of DNA methylation, especially low levels of histone substitution of the acetyl group for a H adapted OH group. Now what is dosage compensation? Ensures an equal expression of genes from the sex chromosomes even though females have two X chromosomes and males have only one. So in each female cell, one X chromosome is inactivated and is highly condensed into a wire body. Female heterozygotes for genes on the X chromosome are genetic mosaics. So X chromosome inactivation, embryogenesis Marie discovered that in female cells, one X chromosome is randomly switched off. This chromosome forms a dense region in the nucleus known as bar body. Bar bodies are generally not found in males because the single X chromosome is still active. This explains why X females do not show symptoms. Inactive X chromosome was present of a heterochromatic bar body in interphase cells. So the XIST gene on one of the two X chromosomes is randomly inactivated by DNA methylation, and the active XIST is transcribed and its RNA product goes to the X chromosome. This suits on the coded X undergo methylation, which causes the chromosome to condense, forming a bar body, and returns inactive. So the uncoded X is left transcriptionally active. Real hypothesis of X inactivation proposed by Mary and Diane Russell. So which X is inactivated? Inactivation of X chromosome occurs randomly in somatic cells during embryogenesis. Now progeny of cells all have same inactivated X chromosome as original, creating mosaic individual. So inactivation is not always random. A structurally abnormal X is preferentially inactivated. Example, isochromosome X, that is extraembryonic membranes that go on to form the amnion, placenta, and umbilical cord. In all the cells of the extraembryonic membranes, it is called as X chromosome that is activated. Inactivation is not complete. Some genes are known to escape inactivation, that is, those with the functional homologue on the Y genes located in the pseudo-autosomal region, still others are specific to X chromosome. Inactivation is not permanent, reversing development of germ cells. In normal female cells, the choice of which X is to be inactivated is random. Females are mosaic with respect to X-linked gene expression. Some cells express alleles or the paternally inherited X, but not maternally inherited X. Other cells do opposite. Females are mosaic with their X-linked genes. Mosaicism is readily detected for some disorders. Mosaicism reveals the random adaptation of one X chromosome, and hypotropic ectodermal dysplasia in a heterozygous woman. Regions where certain lines are absent. Okay. So, what are the chromosomal features of X inactivation? Inactivation of most X-linked genes of the inactive X heterochromatic bar body, late replicating in X phase, exist RNA associated with macro H2 register modifications in chromatin. Synaptation is random, established when embryo is less than 100 cells. Fraction of the cells in career female with normal or mutant allele tend to be variable. The clinical variation in expression of X-linked disorders is common in heterozygotes, ranging from normal to affected. So, manifesting heterozygote in a female in whom the interest of the allele is on the active X in most or all of cells. Okay, X chromosome inactivation, X chromosome translocation will not go into detail. We've already gone through this main concept behind these. So coming to the question that we started with, the sorry, the histone unique to bar body, most commonly associated with the bar body is macro H2A. Okay. This histone is significantly enriched on the inactive X chromosome, which forms the bar body, contributing to its highly condensed and transcriptionally silent state. High concentration studies have shown that the bar body contains a significantly higher concentration of macro H2A compared to other regions of the chromosome. Macro H2A is believed to play a key role in maintaining the inactive state of the X chromosome by compacting chromatin and preventing gene expression. And researchers often use immunofluorescence staining with antibodies against macro H2A to visualize the bar body cells. Okay, so that is the answer for this question. If anybody has any doubt, I'll wait for a few minutes. Then we can conclude the session for today. Okay, there are no questions. Then we can stop the session, conclude the session for today, and we'll meet again next Monday. Thank you, everyone. Thank you, guys. We'll meet again next Monday. you.